Do you ever wonder how our body works? In particular, how are proteins made inside our bodies? All living organisms are based on proteins, from hair to hearts, from skin to vision. All of these are made from proteins. And yes, there are many different types of proteins in our bodies, and they all serve different important roles in our growth, development, and everyday functioning. But what may come as a surprise is that building a protein is a precise process that follows a recipe. Remember our topic about the DNA? Yes, it's the blueprint of life. The code for making a protein is actually written within your DNA, and the message created by the sequence of nitrogen bases in DNA is the message needed to make proteins. Remember that DNA has the ability to duplicate, however, sometimes mistakes do happen, which results in the messages being changed that affects the structure and function of the created protein. These mistakes are called mutation. Now, what do we mean by mutation? Now, let's find out!
Well, a mutation is a change in the DNA sequence of an organism. Mutations can occur in both prokaryotic and eukaryotic cells and can arise in two different ways. First is by failures in the error-checking mechanisms of the cell, or by the changes introduced by the environment such as the radiation exposure. Mutations can also be described in terms of the type of cells being affected. Somatic mutations are those that happen within an organism's body cells during mitosis. The changes in the DNA code get passed from one cell to another when the cells reproduce. However, this type of mutation will only affect the organism itself. It does not get passed on to the offspring. Germline mutations are mutations that occur in gamete or reproductive cells and result in the gametes that have defective genes. Because the gametes are used for a reproduction in a sexually reproducing species, these mutations can get passed on to the offspring.
There are many ways mutations can affect an organism. A silent mutation has no effect on protein function. It may not change the protein that is coded, or it may lie in uncoding regions of the DNA strand. A mutation may instead cause a loss of function, that is, the mutation may cause a gene to not be expressed at all. This changes the phenotype of the organism, its observable characteristics. An example of this type of mutation is albinism, a condition that causes the absence of the skin pigment melanin. Finally, a mutation may be conditional, that is, it may only be activated under certain circumstances. Although this is more common in acellular organisms than in complex multicellular organisms, it can happen in both. For example, sickle cell disease arises when an individual has two copies of the gene for sickle-shaped cells. When an individual has only one copy of the gene, they produce both normal and sickle-shaped cells. They do not necessarily express symptoms of the disease, but the carriers for it. This means they can pass the disease on to their children. There are two types of mutations, a point mutation and the chromosomal mutation.
Mutation that occurs in DNA is called a point mutation. This involves a change in either one or few in the nucleotide bases in genetic sequence, from addition, subtraction, or replacement of the base or bases. This can happen in two ways: base pair substitution and base pair addition or deletion of nucleotides. A base pair substitution is when the mutation causes the nucleotide to be replaced with another nucleotide. When one or more than one nucleotides are added or deleted, it is considered a base pair addition or deletion. Within these two main categories, several different outcomes can occur: missense mutation, nonsense mutation, and frame shift mutation.
When missense mutation occurs, it changes the genetic information or codon so that one amino acid is substituted for another in a given protein and is functionally different from the original. This change usually has harmful effects because of the change in the structure and function of the protein. Another outcome is called a nonsense mutation. This occurs when a nucleotide substitution happens that results in a stop codon being produced. The stop codon is a sequence of DNA bases that tells cellular machinery to end the message that makes a certain protein.
This produces shorter proteins because the translation of the mRNA or messenger ribonucleic acid strand is terminated too early. Like missense, the nonsense mutation can be dangerous to the function of the protein. The last is the frameshift mutation. This happens when one or two nucleotides are inserted or deleted from the genome. This is called the frameshift because the triplet code that is used to produce an amino acid is shifted to the left or right, resulting in a different amino acid being coded. Ultimately, this can result in an entirely new sequence of amino acids. Thus, new proteins are formed and can be harmful to the organism.
In addition to the point mutation that can occur at a single nucleotide, there are also mutations that can affect entire chromosomes, and these are called the chromosome mutations. This happens when pieces of chromosomes break off, get mixed up, and even rejoin in different areas. This type of mutation can cause significant changes in the expression of phenotypes. There are 4 types of chromosome mutations: deletion, duplication, inversion, and translocation. Deletion is the loss of a piece of a chromosome, such as when part of a chromosome breaks off and the two exposed ends rejoin. Duplication is where a part of a chromosome is doubled. This usually results from an unequal crossover resulting in one homologous chromosome having a deletion mutation while the other has a duplication mutation. Inversion is when a segment of DNA breaks off a chromosome, flips around, and reattaches in the opposite direction. Lastly, translocation is when a segment of DNA breaks off and then attaches to a different chromosome.
During the cell's life cycle, errors in the nitrogen base sequence can happen spontaneously. The errors that occur during DNA replication, repair, or recombination are called the spontaneous mutations. This may result from a brief change in the form of the nitrogen base or slip mispairing. When a brief change in the form of the nitrogen base occurs, an isomer is created. If the error-correcting system of the cell fails to repair this error, the DNA polymerase selects a different nucleotide to pair with the isomer during the DNA replication, creating an error in the base sequence. The spontaneous mutation occurs. Unrepaired errors like this can be fewer than 1 in a billion nucleotides per generation. Slip mispairing is the second source of error in spontaneous mutation. This event happens when a misalignment occurs during the homologous pairing of chromosomes. Under normal conditions, cells can revert the misalignment and return to their correct pair. But if the error-correcting system of the cell detects the misalignment and slices the loop, it will delete several hundreds of nucleotides in the chromosome. The deletion of the nucleotides changed the reading frame, resulting in a frameshift mutation. The cells repair damage in the DNA by nitrogen base removal, mismatch repair, and nucleotide removal.
Remember that when the language in the DNA is changed, the language for making proteins is changed too. Mutations can also cause altered proteins to be made that in turn create different expressions of traits. Often mutations leads to problems that harm the organism or even result in its death. This can happen whether the mutation causes a loss of function or a gain of function. Sometimes, however, new traits that arise from genetic mutations are beneficial. In fact, mutations are essential. It's for the evolution to occur as it results in the increase of diversity of new favorable traits among organisms.
So we saw how mutation can change protein messages and create variation. And remember that these changes have been happening for billions of years. They created all living things, from tiny ants to whales and elephants. And as you all know, it's not just our DNA that makes each of us unique. It's also our experiences. So, just like mutating DNA, your next experience could be a life-changing one. So hopefully, you learned something new from this video, or at least have a better understanding of the world around you and yourself. On behalf of Trini Biology, thank you and see you on our next exploration. Goodbye.